So on the functional side of DNA methylation, we know that DNA methylation is very, very important to control gene expression. And so this is coming from a review paper. So people don't actually know initially how DNA came into the genome because bacteria already have DNA methylation. They use it to detect what is a different species or viral infection or other bacteria trying to infect each other, because the DNA from a different species will have a different DNA methylation pattern compared to the DNA methylation in the original species, but they don't always happen even on the C location. In the human genome, there are some hypotheses that, remember, our human genome is 50–60% repeat sequence. A lot of those repeats could be in ancient time, there's a virus that infected human, and they incorporate the viral DNA into the host genome, and that will leave a copy. Usually the virus, if it's a very bad virus, it will kill the host, but then they will never get a chance to live in the host genome. And so the viral genome sometimes incorporates the DNA into the host genome. It can use the host genome to reproduce the DNA, to make RNA, to package additional viral particles to infect other cells. And the host genome has a way to stamp it, to just kind of cover it up and not allow it to be expressed, because obviously you don't want this piece of DNA in the genome. And interestingly, very often when the host genome is not in good shape, the cell is dying or something, then the viral genome has a way to sense the stress, and it was like, oh, my host is in trouble, so it will try to package itself and really leave. But then sometimes it does leave, but sometimes it will just insert itself to another location into the genome. So you will see those repeats in our human genome for the same species that's inserted itself in many, many locations in the genome. And it's certainly not a good thing. And so human genome try to use DNA methylation to really silence that region and not allow that region to be transcribed. And so it's kind of a way for human to deal with potential viral infection and the genome incorporation to maintain the genome stability. And so those are usually in the repeat sequence. And how DNA methylation evolved around the CPG island, that's really something, at least I don't know. Basically, if you look at a particular gene, these are the exons, these are the introns. Very often you will see right in front of the gene, as we mentioned, about 70% of the genes will have a CPG island right in front of this gene. In fact, some of the gene finding algorithms specifically look for CPG island, because if you see a CPG island, there is a chance nearby there is a transcription start site for a gene. And you can use it even as a gene finding approach. And so these regions, in our genome, if you have a C and G, most of the time that CG is methylated. But in front of the, in fact, if that CG is unmethylated, with any conditions, there could be some, so bisulfite, I guess, is just some chemical. There could be other external stimulus which create some little chemicals. The unmethylated C followed by a G is very likely to be just converted to a T. During any cell external condition, it's very likely to be converted. And that's why in the human genome, our CG percentage is lower than 50% compared to AT percentage, is because the unmethylated C followed by a G are usually converted to a TG. So you do see a lot of TG and CA in the genome. It's because of this unmethylated C being converted to a T. Except that in front of the gene, when you have a CPG promoter, there are some transcription factors that really needs to bind to the promoters. They occupy those locations in order for this gene to be activated. And so the CPG promoters can be unmethylated when the gene is poised for transcription activation. And so the unmethylation is because of some other transcription factor occupying that space, not allowing the DNA methylation enzyme to go there to methylate the region. And so that's actually important. And basically in a cell, if the promoter of this region has a CpG island and it's not methylated, it's a potential indication that this gene needs to be turned on in that cell type. Yeah, Patrick.
Because transcription factors like to bind CpG?
So question is whether transcription factor likes to bind to C and Gs. The CpG island that are bound include some transcription factors that really bind in a cell type specific way, but also the whole polymerase complex is binding to that region as well. And the CG is actually good for the whole transcription complex to assemble in front of a gene. In the distal region, on the enhancer regions, usually you don't have a CG island. You just have a single CG occasionally on a transcription factor binding site. And depending on the transcription factors, so far what people have found is that some transcription factors, they can only bind to the non-methylated CG. Even if there's a single CG that's methylated in this enhancer region on the motif, or even nearby, the transcription factor cannot bind. However, there are some other transcription factors that favors the binding of methylated C even better than the unmethylated C. And some of them actually specifically go there to recruit either the methylation enzyme or the unmethylation enzyme. Right, so basically, some of them can bind to both the methylated and unmethylated with slightly different affinity, but once they go there, they either recruit more methylation or they recruit more demethylation to even, so you can think about this as if this region is DNA methylated, is locked up. And so some of the transcription factors can only go to a region when it's open, but other ones can go to it when it's kind of halfway locked, in order to either lock it further or open it. Right? So that's very interesting in terms of gene regulation. For most of the transcription factors, they prefer to bind to unmethylated regions. Okay. Yeah. So if you look at the promoter regions for the genome, it's usually unmethylated, especially if that gene needs to be expressed in the cell. And we mentioned before, some genes can be imprinted, so the mother copy and the father copy can have different DNA methylation. This is another study which is also kind of interesting. They didn't use bisulfite sequencing, but the result is kind of interesting. You look at this gene, this is the this is the beginning of the gene. It has this very strong CpG island. But in here, you can see there are some other exons that also have CpG island in here. But it's not as big as the first CpG island. It's kind of weaker. And these are also kind of exons. And what people observed is that very often, genes that are highly expressed have CpG methylation on their gene bodies, especially high on the exons. You can see it's really high in here, and it's also really high in here. But on the promoter, on the real transcription start site, this region is unmethylated. And so you're wondering, why does it have, if DNA methylation is supposed to silent binding and gene expression, why do we see this type of DNA methylation pretty high in the gene body, especially enriched on the exons? So this particular Nature paper suggests that sometimes the internal exons could also start transcription. So a lot of these internal exons can also have a little transcriptional signal. So the cell can sometimes use this as a transcription start site, as the first exon, but sometimes it can also use that internal exon as the beginning of the transcript and start transcribing from here, especially if this region also have a CpG island. Therefore, having the DNA methylation in those locations, make sure the cell knows where to really start. Because if this CpG island is methylated and this CpG island is methylated, the transcription start site will not go there. It will only start from the unmethylated promoter to transcribe. And once it starts transcribing, it will just keep going. Having methylation here will not stop polymerase from reading through and continue the transcription. So sometimes having the DNA methylation in those exons, especially on some, actually on many highly expressed genes, you will see that the methylation is pretty high on the gene body, potentially as a way to suppress those regions from starting transcription in the middle of the gene. Okay, so that you can see, in some sense, epigenetics come around because, at least in mammalian genome, because our genome is so big, when the transcription factors or polymerase go into the nucleus, is trying to make some decisions, where to bind, where to start a gene. Epigenetics stands as a landmark to tell them where to go and where not to go. And this is just an example for that. There are also other recent studies showing that in some really important genes, you see you can see in both these examples, in the promoter and also even in front a little bit further, about 10 kb surrounding the transcription start site, you would see this big, well, they call it canyons because it's not just, usually CPG island is only one, yeah, you can see here, the CPG island size is only this big. A few hundred base pair to a kb, but when you see these canyons, it can be as long as 10 kb, and in this region, DNA methylation is much, much lower. And they are very often in the important transcription factors in the cell. This one, they didn't draw the later part of, I would suspect the later part of this gene are actually methylated because the transcription factor is important. It needs to have a pretty high expression, and you don't want to start at the wrong place. And so the later part of this gene could be methylated. But in the promoter, not only on the CPG island, but like quite close to this, you could have a 10 kb region that's unmethylated. And in those cases, very often, I would suspect that there are more transcription factors really binding there, which help this gene to be well maintained for its transcription. And so they look at those regions that have these DNA methylation canyons, and you look at their gene ontology analysis. These are embryonic morphogenesis, homeobox, transcription regulation. So these are important genes that need to be tightly regulated. And in those genes, you very often see some other histone modifications, which we'll talk about next lecture, but suddenly very, very low methylation. That indicate that this gene is potentially important. Okay, so you see another landmark that can tell you whether something is useful. And in diseases, we see some interesting trend on DNA methylation, especially in cancer. So we mentioned that a lot of the genome, especially repeat region in the genome, are mostly methylated. This is kind of globally, a lot of the broad regions are methylated. Interestingly, in the cancer, you see them being a little bit hypomethylated, which means less methylation. And so this could potentially mean the cell is losing its ability to make a control. Those regions that need to be silent is no longer being correctly silent. And so in fact, in cancer cells, you see a lot of these repeat sequence now being activated. They are making viral RNAs are being transcribed now. In addition, there are some regions that are normally methylated. Those genes need to be suppressed. In their promoters, they are usually methylated, so the gene is not So in the promoters of some genes that normally is unmethylated, that you need this gene to be on, but sometimes in cancers, they are becoming silent. And then occasional regions, you see this hypermethylation on some focal regions. And so the corrected genes that need to be expressed in the cell are now being silent. For example, it could be a tumor suppressor gene that suppresses the tumor from uncontrolled growth, but if it's silent, then the cell can continue to grow. And so basically, these type of genome-wide or global hypomethylation and the focal CPG island hypermethylation is kind of a hallmark of cancer. And it can be also used for early cancer detection. Imagine if in your blood, if you if you take the cell-free DNA in our serum, because all of our cells normally die and it will shed some DNA into the blood. There are a lot of regions that should be unmethylated, and globally in these regions, they should be methylated. But then suddenly, if you in the blood, you take the DNA that's not in the cells, it's just shed the DNA from wherever in your tissues, suddenly you see a lot of these repeat regions that are being now unmethylated, and those that are supposed to be open are now being methylated. That's actually a potential way to look at early cancer. And sometimes based on which regions become methylated, you can even guess what cancer it is. And so there are active research in the area. It's not, there are papers published. It's not really developed as a clinical assay yet, because you don't want to get false alarm too often. The specificity of such assays have to be super, super good. But I think the technology and also the bioinformatics are both being developed around this. There are also other studies showing this is also in cancer and sometimes early cancers. This one is not on the blood, but on the tissues. You can look at the DNA methylation. Some of the regions, so you can see in the bottom, these are, so this is a 1 kb, this is where a CPG island genes are. So these genes have CPG islands. So there's a density of good genes, and on their promoter, these regions are unmethylated, meaning that these genes are active. Whereas these genes in this cell are usually methylated, so they are not expressed. And then sometimes in cancers, you will just see these regions that are on the, so very often you see these are called the CPG island shores, which means you can have a region which is fairly gene dense and have a lot of CPG promoter genes. And then there are other regions that are kind of CPG poor, and they are usually silent with DNA methylation. And on these shoulder regions, from the CPG poor to the CPG rich or the gene rich regions, suddenly on these boundary regions, you see a difference in the cancer and normal. This is another potential indication. The cell, so basically, normally, if the cell physiology, everything is right, the cell should know very well which regions need to be active and which region needs to be inactive. It's very tightly regulated and very actively regulated. But in cancer, when you see something like this, it means the cell is kind of losing its control. It doesn't know where to turn on or off. And so in this region, not only you see a difference between cancer and normal, you see a big variation. The cell is just can't control its boundaries anymore. And people are also using this type of approach, for example, to look at cervical cancer. For example, with cervical cancer, you can have a pap smear. You take the cells from the epithelial cell in the cervical tissue. And sometimes even by pathology, the doctor cannot see as big a difference. Sometimes from DNA methylation, they can already see the cell are starting to lose control of its DNA methylation. And that's also a good way for potential DNA tumor detection. Okay. And we mentioned that DNA can also be unmethylated. In fact, if you look at where DNA methylation or demethylation really happen, in a normal cell, not necessarily in a disease. If you look at so there are some technologies developed. It involves a very sophisticated biochemistry followed by sequencing. People have done a handful of those mappings. This is definitely not done for a regular profiling approach. There are a few labs that can do this type of experiment, and once they map the hydroxymethyl C, remember this is about only 1% of all the CGs in your genome, a tiny percent, and that's hydroxymethylated. So hydroxymethyl C is already less than the cytosine and the 5-methylcytosine, and formal C and carboxy C is even lower in terms of abundance. And when people map where these hydroxymethyl C happens in the genome, they saw that they are actually really enriched in where transcription factor binds. And so basically, very often, in order for a transcription factor to bind to a location, that CG needs to be unmethylated, actively unmethylated by the TET enzyme in order for the correct transcription factor to bind to these locations. In fact, the TET family of protein are very, very important for DNA demethylation, and when they are mutated, there are very well-known cases of leukemia cases when TET is mutated. Then DNA demethylation cannot happen, and that's pretty bad. Okay, so that's DNA methylation. So basically, you can think of DNA methylation as a general way for regulating gene expression, mostly as a suppressive mechanism. When it covers the gene, when it covers the repeat sequence, those viral DNA can no longer be activated to make a wrong transcript that the human genome does not need. When it's covered on the transcription factor binding site, very often a transcription factor would not be able to bind there. When it covers the promoter, that gene will not be activated. If this gene needs to be activated, that promoter needs to have low or no DNA methylation on the CpG island of that promoter. And if it's high on the gene body of a gene, it usually indicates that that gene is actually fairly highly expressed, and the methylation there is to make sure that the transcription start site the transcription does not start at the wrong place. Okay, so that's kind of the general function of DNA methylation, at least the current understanding of that.